Chères téléspectatrices, chers téléspectateurs, je suis très heureux de vous retrouver dans votre émission وحضارة, Sciences et Civilisation. Aujourd'hui, nous allons voyager dans le domaine de la biologie moléculaire et de tout ce qui concerne les possibilités de guérison du cancer. Je suis très heureux d'accueillir mon invité, c'est Abdeladim Moumen. Bienvenue dans l'émission.
Thank you very much.
Vous avez, donc je viens de le dire, votre spécialité c'est la biologie moléculaire, mais il est de tradition dans cette émission d'abord de parler de votre formation, de votre motivation aussi, pourquoi vous avez commencé ces études-là que maintenant, qui sont devenues votre profession. Vous avez grandi dans quelle ville au Maroc?
Actually, I was living in Casablanca before going to France to perform my PhD. So I have spent my university studies here in Morocco, in Casablanca University, before going to France and specifically to Nancy University, in which I have performed a master degree. And after that, I joined the Pasteur Institute as a PhD student.
L'Institut Pasteur, très connu, bien sûr,
in
pour
Paris,
toutes les études de virologie.
Absolutely. And this is why actually, because that was at that time, that was a lot of research on HIV. And this is why I was really interested.
Dans les années 2000, donc.
I started my PhD in 1997. 1997, 98. So I started my PhD and I was working in the molecular biology department in the Pasteur Institute under the guidance of Professor Henri Buch, who was actually, why I'm saying his name, because he was also a student to a big name, who is the Jacques Monod, who was the father of the molecular biology actually. Oui. So I was really honored.
Donc vous étiez dans un entourage des grands pères de la biologie moléculaire, mais adapté à cette réalité de contexte, une réalité malheureuse du virus du sida, et tout le monde travaillait dans cet horizon. Et vous-même, vous avez commencé votre thèse dans cet Institut Pasteur en France, à Paris, autour de la virologie?
Absolutely. So my project was actually to dissect the mechanism, molecular mechanism, on the genetic variability of HIV. You're working on a homologous recombination. You know that the virus, HIV virus, is a retrovirus that contains two molecules of RNA, which constitutes his genome. And during the reverse transcription, because the first step of infection, the RNA has to be changed and to convert to DNA, and this is what we call reverse transcription. This is the opposite of transcription, which is going from DNA to RNA, and this is going from RNA to DNA, called reverse transcription. And during the reverse transcription process, the reverse transcriptase, the enzyme who performs this reaction, can switch between the two molecules of RNA. And in a region which is homologous between the two RNAs, this is what we call homologous recombination. It's a kind of strand transfer during the synthesis of the first strand of DNA and going from an RNA, going to another one. And this will increase the ability of the virus to change its genetic and to generate other strains which are more resistant to other drug. And this is important to study just in order to know how it works and to find way to inhibit it and to block the variability.
C'est très intéressant. Donc déjà à l'époque, on travaillait sur les vulnérabilités génétiques et les instabilités génétiques qui ont été précisément, qui sont responsables de l'accueil malheureux de cette maladie et qui fonctionnent, comme vous l'avez dit, de manière inverse à la transcription normale des gènes. Et là, c'est précisément un élément de rétroversion de l'ARN vers l'ADN et non pas dans l'autre sens. Et sur cette rétroversion, votre travail de thèse consistait en quoi?
This is the studying the mechanism by which the reverse transcriptase can go from a molecule of RNA on which it starts the reverse transcription to another one and changing the way. And it could copy one piece of the first molecule of RNA and it could carry on the other piece of another molecule of RNA, which could have a genetic variability which is different. And it could give a hybrid of DNA containing, for instance, two resistance to two different molecules which have been used to treat the HIV. And this is a problematic because we will generate strain which are more resistant to drug. So this is how the virus could escape from therapy and from the immune system as well.
Il est d'une malice incroyable, le virus,
Absolutely
le virus du Sud.
Fantastic
Et
ability.
il faut toujours assez le suivre. Et donc, dans votre domaine génétique, il fallait tracer les voies qui permettent de suivre la malice de ces contournements. Et ça, c'était votre premier travail.
This is the first thing that I've done. So I have developed a system, what we call an in vitro system, by which I can study the homologous recombination in HIV and retroviruses in vitro. That means in tubes. And after that, I generate another system using cells by infecting them with I could say artificial viruses that contains everything that I want to study on. And this is just to know how the reverse transcriptions homologous recombination occurs and how can we inhibit it.
La connaissance de ces chemins labyrinthiques du virus se fait par des simulations.
Absolutely.
Et donc,
artificial matrix.
Voilà, qui sont donc des éléments artificiels qui permettent de modéliser.
To reconstitute what happened actually in the cell in a very real time.
Voilà, et donc pour mieux savoir ce qui se passe dans cette cellule, on fait cette modélisation, cette simulation qui permet de performer la connaissance, de mieux générer la connaissance de ce qui se passe in vivo, alors que là c'est in vitro, de cette façon. Là c'est tout à fait fascinant. Après tout ce premier travail, vous rejoignez le Royaume-Uni. D'où votre magnifique anglais et avec un accent so British.
Thank you very much.
Et là, vous rejoignez deux univers. Je dis deux univers, mais vous allez me dire la connexion. Un travail à Cambridge University et un travail à London University. C'était quoi précisément qui vous a mené de l'Institut Pasteur à Paris à rejoindre précisément le Royaume-Uni et particulièrement ces universités-là?
Absolutely, this is a very good question actually, because I was kind of prepared to go to the UK, and because as I've said, I was working on the reverse transcriptase, but in the same laboratory in which I was in PhD, during my PhD, there was a lot of other research. I was working on other things. And one of them used to work on the DNA damage response. And this is quite different from retrovirology and molecular virology. So I was in discussion with this guy. I was quite attracted to this field because this is a field that could make me understand very well how can we treat cancer. And that was another chance. So I changed completely my field of work and research when I went to Cambridge. So via this guy, I've got a big name in Cambridge, who was one of the famous laboratory in this field of understanding response of the cell to DNA damage. That was the laboratory of Steve Jackson in the Cambridge University. So I have applied for a postdoctoral training there. And I have been accepted. Fortunately, I was very happy.
Et oui, parce que on le dit très souvent dans cette émission, le destin d'un chercheur, c'est de se déplacer. Mais parfois, c'est la recherche elle-même qui se déplace. C'est-à-dire que vous avez obtenu ce postdoc au Royaume-Uni et ce postdoctorat, donc les études qui commencent après le doctorat, qui inscrivent complètement le chercheur dans sa recherche, sur le long terme, on the long run.
Yes.
Et sur ce long terme, ça commence précisément par ces études postdoctorales, disais-je, et vous êtes accueillis dans un univers où vous, et c'est aussi le hasard et les opportunités de la recherche,
Yes.
il suffit d'un déclic de conversation
Absolutely.
pour que l'on se dise je vais aller regarder de ce côté, et là c'est la réponse cellulaire à des dommages sur l'ADN. Donc on n'est plus tout à fait sur le problème entre l'ARN et l'ADN, et vous vous dites bon, j'étais en virologie mais maintenant c'est peut-être le cancer, c'est certainement le cancer avec ses incidences moléculaires que je vais analyser.
Yes.
Et vous avez raison d'insister sur la figure des personnes que l'on rencontre. Vous avez cité ce collègue qui vous accueille, ce domaine qui était bien structuré, parce que je suppose, et c'est ma question, qu'on change aussi d'univers de recherche quand on voit un univers très structuré qui est capable d'accueillir une recherche qu'on va faire ad initio, de nouveau, complètement.
Yes.
C'est-à-dire que si vous avez trouvé quelque chose de pas très structuré, vous n'aurez peut-être pas été de ce côté-là.
That would be very difficult to get in the Cambridge University, and mainly in this laboratory, because this laboratory is, as I have said, was a famous laboratory in this field, and you have to have come from a big lab in order to integrate it, because there was a lot of, asking for joining this lab. There was, we were actually in this lab something like 19 postdoc, imagine. So this is a huge lab in which there is different pathway of the DNA damage response that you can imagine that was studied. And in this laboratory, most of the protein which are involved in the DNA damage response has been discovered. This is very important. So it's a
Donc ça lui donne de l'autonotoriété dès lors
very
qu'il y a eu déjà des résultats.
Yes.
Parce que une des grandes problématiques de la recherche, c'est du point de vue social, c'est de dire on finance la recherche, mais quel est l'usage thérapeutique, qui n'est pas toujours évident au début, parce que la recherche est imprévisible, on le dit très souvent, on ne sait pas quel sera l'usage thérapeutique. Et vous dites que ce laboratoire qui vous accueille à Cambridge University avait déjà eu des résultats thérapeutiques qui lui permettent d'avoir des moyens budgétaires et pour continuer précisément à attirer. Vous étiez 19 postdoctorants, it's huge, c'est beaucoup de monde.
Very competitive as well.
Et bien sûr.
Because when you are going to a laboratory like the one I was in Cambridge, you have to be very competitive. You have to publish in high school journals, actually, because they have the habit to publish in Nature, in Science, in Cell, and this kind of journals. The requirements is really huge and big, and you have to have very solid projects.
Alors, est-ce que cette compétitivité, vous l'avez bien vécue ou est-ce qu'elle est stressante? C'est-à-dire de devoir être au top de la recherche was it exciting? Est-ce que c'était intéressant pour vous de vous mettre dans ce combat?
Very interesting, because I'm a person that like challenges. So I like to get challenged. This is why I want yes, this is why I wanted to change, actually. So I want to change from virology to cancer. And this is big. Even though we use, we use molecular biology, genetic, but it's not the same way.
No.
In cancer, you have to use a lot of cells, a lot of in vivo studies, ex vivo studies. It's not only tube studies, in vitro. And also the laboratory was competitive, so I have to accept this challenge. But it was competitive, but not stressful. This is the beauty.
Ça, c'est très important.
And this is because of the guy who lead the lab.
Ah, bien sûr, ça dépend des tempéraments, parce que l'on soit compétitif sans rivalité, que l'on soit compétitif sans stress, c'est la recherche de laquelle on peut rêver. C'est-à-dire que les intelligences contribuent sans exclusivité, sans stress psychologique. C'est magnifique. Et donc, dans ce nouveau champ qui est le vôtre maintenant, la cancérologie à partir de ce postdoc, vous avez continué dans ce domaine. Alors, il y a malheureusement plusieurs types de cancers. Et vous vous êtes beaucoup intéressé au cancer de la prostate, au cancer du sein.
So the main project used to be is to understand how cell responds to the DNA damage which is provoked by the therapy used for the cancer, any cancer. You know, there is two main therapies which are used and people know is the chemotherapy and radiotherapy. These two methods are triggering DNA damage in order to kill the cell. But, you know, this DNA damage is always used in the cell. We can have it while we are normal. So we have DNA damage around us. We have UV light. We have molecules that we ingest during our foods. We have a lot of things that we have inside our cell that could damage the DNA. But our cell are equipped with a system that will arrest the cell cycle in order to repair. And this is the beauty of the cell. So they can repair the DNA damage in order for the cell to carry on with this division. But if we can't repair the DNA damage, we will kill the cell via a system which is called apoptosis. And apoptosis is a kind of suicide of the cell because we have a lot of damage in DNA. What we do with the treatment of cancer that we generate a lot of damage in order to kill the cell, to lead it to the apoptosis pathway. But sometimes cancerous cells, because they are very random cells, they multiply in a very random way, they will generate a lot of mutation, they will not care about the damage. They can carry on with a lot of damaged DNA, which will make things worse for the cancer. This is why we have to understand how cells respond to DNA damage, how it could repair it, and how can we inhibit this ability to repair the DNA damage in cancer cells, because we want to kill it.
Oui, ce qui est véritablement fascinant, c'est comment vos recherches prennent acte du fait que la chimiothérapie et la radiothérapie endommagent même les bonnes cellules, mais la cellule elle a sa propre intelligence, elle sait elle-même lutter contre ces dommages-là, et vous voulez renforcer la manière qu'a la cellule de lutter contre les dommages de la chimiothérapie et de la radiothérapie.
So the idea was behind to find out a way by which we can enhance the effects of radiotherapy, radiotherapy and chemotherapy, and to make them more efficient as they are. And this is why I was working, I used a technique which is called proteomic analysis, just to check what kind of protein could be changed in their structure, in the function, following DNA damage, and to choose one and to study it. And I was lucky to discover a new pathway.
Magnifique.
That have never been discovered before.
Toujours à Cambridge.
It's always a Cambridge University. So, and this pathway is governed by a protein which is called HNRPK, which used to be known by another function, but never been known by its function on the DNA damage. And I was the first to show
Nouveaux usages.
New function of this protein.
C'est ça.
So, and this protein will work with another protein which is already known, which is called P53, and this is a very well-known protein in terms of DNA damage response, because this P53 is very important to stop the cell cycle once the DNA is happening, and after that, if there is too much DNA, P53 itself will generate apoptosis.
Alors, on résume. Après la virologie à l'Institut Pasteur en France, et autour des travaux sur le virus du sida, après le moment de Cambridge où vous impliquez complètement dans la réparation des cellules, l'autoréparation renforcée, la cellule s'autorépare contre les dommages de la chimiothérapie et de la radiothérapie, et vous les renforcez, vous avez découvert le moyen que les cellules ont de lutter contre ces dommages-là. Vous voilà à Londres.
Voilà. So in London University, I've been
assistant professor,
associate professor. First, I started assistant professor, promoted to associate professor, and a lab head also, because when you are an associate professor in the University of London, you got a lab, and you are a lab director as well. So I've got students, postdoc, and I set my own lab. I set my own lab in the London University.
Oui, magnifique.
Yes, that was absolutely something. It's another challenge as well, because now you don't do research for yourself, but you're leading
vous organisez la recherche des autres.
You have to lead the research, and you have to have this leadership mechanism.
Et quel était le label de votre laboratoire à Londres?
I continued on the same specialty of DNA damage response, working on other pathways and discovering other proteins. And actually, with my postdoc and students in the lab, we've been able to publish a lot of work dissecting the mechanism by which DNA damage could be responded by the cell. And,
il y a beaucoup d'espoir autour de ces cellules finalement qui luttent efficacement contre les dommages.
We have jointly with a laboratory in the Karolinska University in Sweden, in a collaboration, we have found out a new molecule that will enhance the efficiency of the chemo and radiotherapy by inhibiting a pathway that we have discovered in Cambridge and in London.
Est-ce que ça va de pair avec ce que l'on dit aujourd'hui beaucoup des thérapies ciblées?
This is it. Actually, this is a kind of molecule that we can give in the same time as chemo and radiotherapy to enhance their effects. So the cell will not be able to repair the damage that will be provoked by these two methods of therapy. And the cell will die. And this is what we want. We want to kill the cancer cells. So we have to give them something that fragilizes them and that they will be unable to respond efficiently for repairing the DNA damaged and provoked by the therapies. And the therapies will be more successful and will kill cells.
Faire quelque chose de plus étanche entre les cellules malignes et les bonnes cellules.
Yes. And we used to work also to combine, as you have said, target therapy by using monoclonal antibodies that will target our molecule specifically to a cell. For instance, if we talk about breast cancer, for instance, we have proteins which are mainly overexpressed in breast cancer.
On a fait beaucoup de progrès contre le cancer du sein.
Yes, breast cancer, that was because there was a lot of money put on the breast cancer.
Et oui, ça va souvent de pair. Le cancer de la prostate aussi, vous y êtes intéressé?
Yes, at the moment, prostate cancer is something that is, very interesting to work on.
Beaucoup d'espoir thérapeutique.
And when I came back to Morocco,
ah oui, maintenant c'est la dernière phase.
Yes. So after passing spending something like 10 years in England, I've got a prize here in Morocco by the government and the Ministry of Research here in Morocco in 2009, and I've got the second prize. That was a big prize, which is governed by this ministry and the industrial ministry as well. At that time, he was Mr. Ahmed Rida Shami, who used to be the Minister of Industry.
Félicitations.
I met him. And he has talked to me about the Masir Foundation.
Ah, Masir Foundation, il faut absolument évoquer.
Yes, that used to be, it has been created actually in 2007, if my memory is good. And he was introduced to me, this kind of foundation, that it is interested mainly in research and development.
Voilà, Fondation Masir, la fondation marocaine, pour la science avancée, Advanced Science, et c'est là que vous la rejoignez, et d'ailleurs c'est là que vous êtes aujourd'hui dans cette fondation qui est maintenant hébergée à l'université Mohammed VI Polytechnique.
Part of the UMCSP.
Avant elle était dans le domaine ministériel public, et maintenant
vous êtes
in
the ecosystem of UMCSP actually.
Voilà. Et le travail que vous effectuez maintenant là-bas consiste en quoi? Est-ce que c'est en continuité avec?
No, absolutely no.
Plus du tout.
This is absolutely different because I was also challenging myself now.
C'est magnifique.
And to go through now research and development. Research and development is different from fundamental research that I used to do in England and in France. Fundamental research is to understand mechanisms in order to find way to fight against cancer, against virus, something like that. Research and development is to use research, results from research.
Vous êtes plus dans les sciences appliquées maintenant.
Yes, and to develop products.
C'est ça.
So developing products is another challenge.
Les kits en particulier pour améliorer les diagnostics.
That's it.
De quoi s'agit-il?
So I have been in a laboratory that develops kits for diagnostic for several diseases, such as cancer, and
mais aussi
infectious diseases,
des infections,
infectious diseases, using molecular biology methods, among which the PCR. PCR is a way by which we can amplify the DNA. So if we have a small amount of DNA, we use this technique to amplify it. Two methods of amplification. One could be end amplification that we can see on a gel, and another one which we call real-time PCR. so we can follow the reaction while it is performing. So with a curve that we can see on an image, and we can also quantify the DNA or the RNA that we are amplifying. And this is very important because we can amplify any RNA coming from a virus, coming from bacteria, coming from a cancerous cell. And
moi, je trouve que vous unifiez vos recherches.
This is it.
Cette nouvelle étape marocaine permet d'unifier les recherches fondamentales.
I use my expertise as a molecular biologist
sûr,
in order to develop kits which will quantify protein for specific cancer. For instance, we have a kit for breast cancer, HER2 overexpression breast cancer, that will quantify the protein HER2, and it is very important for the pathologists and for the oncologists because they have to know whether the breast cancer is overexpressing this protein in order to give the lady who is affected by this who is affected by this disease, breast cancer, a very specific molecule to fight against this breast cancer specifically. And also we have another kits for leukemia that we have released recently, and it could follow up the disease throughout the treatment. So once we are leukemia, we give a specific drug, and we have to follow up the disease in order to know whether the treatment is efficient or not, and whether we can change it, arrest it, or carry on with the disease.
Le suivi moléculaire des patients.
And now we switch also to infectious disease, and we work on the hepatitis C.
Ah, ça c'est très important, l'hépatite C. Ma question sur l'hépatite C, c'est comment améliorer la prévention.
Absolutely. So our first kit that we have developed for hepatitis C, that was just to follow up the disease. We have a very efficient drug for hepatitis C at the moment, and we need to know when the patient is completely free of the virus. And how can we know that? Using our kits. Our kits will give you exact number of virus in a specific sample. So we take a sample from the patient, and we can measure the quantity of the virus which are present in this sample. And after giving the patient this drug, we can ask him to return or to come back in 6 months, for instance, or 3 months, and we re-perform another test using our kits in order to measure the quantity of the virus now. And we will see whether there is a decrease in the virus or there is a resistance to the drug or something like that. And this how we can by this way we can follow up the disease, and we can know exactly when we have to stop the treatment and when we have to say to the patient that he's free of the virus.
Merci beaucoup. Nous arrivons au terme de l'émission. Merci du moment de recherche fondamentale commencé en France et à Cambridge au suivi précis des patients dans leur thérapie. Merci beaucoup.
Merci beaucoup.
Si Abdeladim Moumen. Merci, chères téléspectatrices, chers téléspectateurs, pour votre attention et votre assiduité à cette émission et pour cet échange très passionnant que nous eûmes ensemble. Merci, Monsieur Moumen.
Merci beaucoup. Merci.